Breast cancer affects around 1.5 million women worldwide. While earlier detection and better standards of care have increased response rates, many will have their breast cancer recur. Thus, new approaches are needed to prevent disease recurrence. Despite attaining remission with standard of care, residual cancer cells can remain. These cells can travel through the circulatory system and migrate throughout the body. Here, one or more breast cancer cells have migrated to this bone site and has begun growing. If not stopped, this micrometastatic nest of cells will grow until it is able to be diagnosed as a metastatic tumor mass. HER2 is a protein product of an oncogene that is highly overexpressed by approximately 85% of breast cancer cells. In vitro and in vivo studies have identified the NeuVax peptide sequence as being the most immunogenic fragment isolated from the extracellular domain of the HER2 receptor. NeuVax is injected with a simple intradermal injection along with an immunoadjuvant. Upon injection, localized antigen-presenting cells, known as APCs, pick up NeuVax on specialized surface receptors. These loaded APCs then migrate to the lymph nodes, where they bind to T cells with specific HER2 receptors on their surface. Upon binding, a rapid expansion of T cells specific for HER2 expressing proteins takes place. Millions of NeuVax-specific T cells leaves the lymph nodes and circulates throughout the body seeking out HER2-expressing cells and micrometastases. Upon binding of these NeuVax-specific T cells to the HER2-expressing tumor cells, the T cell neutralizes and destroys the tumor cells. Clinical data has indicated the ability to maintain a long-term elevated level of NeuVax-specific T cells, potentially providing long-term protection against the possibility of a tumor recurrence. For more information, please visit NeuVax.com.